Welcome to the WebEVTalk series. This is talk series for EV enthusiasts around the world. So today is the 6th July 2021, and it's my pleasure to welcome Marcio L. Rodrigues from Brazil. So Marcio has been working on fungal physiology and pathogenesis in Brazil for the last 20 years. And his group described the fungal extracellular vesicles for the first time in Cryptococcus pathogenic model in 2007. And then since then, fungal EVs were characterized in 15 genera by a number of groups. So today, his lecture is going to be about insights into us. Please go through patreon.com and look for us, and you can support our activities through Patreon. So please welcome Marcio. Please go ahead, Marcio.
All right. All right. So thank you so much for this invitation. It's a pleasure. It's always a pleasure to talk about fungal extracellular vesicles. I really actually appreciate this kind of space because since this is a relatively new field, we are always fighting for space, for visibility, and to talk about this kind of models, it's really important for us. So Carolina, thank you so much for this invitation. It's a pleasure. And I'm going to talk about some of the insights into the analysis of functions, the analysis and functions of fungal extracellular vesicles. This is the kind of work that we have been doing in Brazil. And in general, I'm going to talk about fungal biology and EVs, the cycles of biogenesis of fungal EVs, and some of the recent functions that have been described for fungal EVs. But before, just to give you a general analysis of where we are, let me talk a little bit about our institution. We are the Oswaldo Cruz Foundation, Fiocruz. Fiocruz it is actually a system in Brazil. You can find units of Fiocruz all over the country, but we are the South unit of Fiocruz here in the city of Curitiba in the state of Paraná. This is our institute, the Carlos Chagas Institute, and this is the Fungal Infections group. Actually, a big part of my work in this field has been developed in the city of Rio de Janeiro, where we were until 3 years ago. And we started a new group 3 years ago in the Institute, and this is us. This was a very special day for us, the day that we got vaccinated 1 month ago. I'm going to talk about fungal diseases. So, of course, the focus is extracellular vesicles, but our model is fungal infections. And this kind of disease, they are really important for human health. Every year, more than 1 million people die because of fungal infections all over the globe. And our main model, it is actually the most important fungal killer. It is a genus called Cryptococcus that causes a disease called Cryptococcus. Cryptococcus is a lung disease that can evolve to a brain infection in immunocompromised patients, and it is really deadly. Cryptococcus kills almost 200,000 people every year, and if this kind of disease is not treated, it is 100% fatal. So it's an important problem of human health, and we have been working with this model for I don't know, 2 decades. And Cryptococcus, of course, is not so common compared to other diseases, but you have an additional problem in Brazil, at least. The treatment of Cryptococcus is really, really expensive. Per patient, it costs more than $100,000. And for example, in Brazil, we have approximately 7,000 patients. If you give the right treatment to all of them, you're going to spend a lot of money, and this money is not available in the public system in Brazil. We have been trying to find, to get more visibility to fungal diseases, but it's not easy. In general, this kind of diseases, they're really neglected. And I think it is time to stop neglecting fungi because, it is a big problem of human health. You don't have many options to treat fungal infections. You have no licensed vaccines. So it's important to take a look at fungal infections and to find new tools to fight these kind of diseases. And today I'm going to talk about, of course, Cryptococcus, but I'm going to include 2 additional systems. So I told you about Cryptococcus before. I'm going to talk a little bit about Aspergillus and Aspergillosis, I'm going to talk too about a plant pathogen, which is Penicillium digitatum. So I'm going to use these models to illustrate the functions of fungal EVs, and each of these models is it's going to be important for the illustration of one specific function. But of course, I'm going to talk more about Cryptococcus, which is our main model of investigation. So let's start talking about fungal biology and EVs. And once again, I'm going to use the Cryptococcus model of secretion. This is really important because Cryptococcus, like many bacterial cells, it is surrounded by a thick capsule. So this kind of structure that you're seeing here is the capsule of Cryptococcus. It's composed of sugars, and it is really important for disease. Without making this kind of capsule, Cryptococcus is not able to cause disease in the human host. But besides the capsule, most of the virulence factors produced by Cryptococcus, I mean, the molecules that are important for disease, they are actually extracellular. So secretion in Cryptococcus is really important for disease progress because, once again, most of the factors produced by Cryptococcus, to cause disease, they go outside. And once again, the capsule is really important. It is a big network composed of different sugars. The structure, it is really complex. I'm not going into the details, but it is a polysaccharide composed of mannose, xylose, and glucuronic acid. So this is glucuronic acid, this is a mannose backbone, and this is xylose. Because of this composition, this polysaccharide is called glucuronoxylomannan, and I'm going to abbreviate using GXM, which is the most common name for this polysaccharide. So GXM, the most important capsular component, is synthesized inside the cell, and it goes outside to form the capsule. And this kind of process is really, really efficient. For example, if you take a look at the capsular area, the concentration of GXM, the polysaccharide, is really, really high. For example, in this kind of region here, the capsular area, the concentration of GXM can reach 10 milligrams per ml. It is really, really high if compared to other biological systems. Actually, during infection, you can find a lot of polysaccharide in the serum and plasma and urine of the patient. So to go outside, you need a really efficient mechanism of transport of the scapular polysaccharide, once again, which is important for disease progresses. So if you think about secretion in eukaryotes in general, this is actually the general model. Things are synthesized inside the cell, they go to the cell surface, and then the secretion process is complete. But if you think about fungal cells, you have a very important additional layer, which is the fungal cell wall. So everything that is synthesized inside the cell and goes outside, it needs to cross the cell wall. This is a very important barrier. If you take a look at this picture here, you can tell that the cell wall is really thick. So everything that comes from here, it goes outside. It is necessary to cross the cell wall. So there's an important question in the field, which is related to the final steps of secretion in fungi. So how do they cross the cell wall? And the answer, like in many other systems, plant cells, gram-positive bacterial cells, it is extracellular vesicles. So we're going to talk a little bit about transcell wall secretion in fungi. And this is actually a field that started 20 years ago when we detected lipids on the cell wall. So this is the fungal cell wall, and this is staining for a lipid called glucosylceramide. I'm not going into the details, but we found a lot of lipids on the cell wall, and they were not expected there because lipids in general, they are found in the plasma membrane. When we saw lipids on the cell wall, we started to suspect that they could be transitory components crossing the cell wall. And maybe they were components of vesicles, structures resembling vesicles like you see here, crossing the cell wall to go outside. Indeed, 5 years later, during the exploration of this kind of hypothesis, we used fungal supernatants to isolate vesicles, and we saw a lot of them there in the Cryptococcus model. So this is the first picture that we had about showing EVs in fungal cells. And actually, these vesicles were full of polysaccharides. So GXM, the polysaccharide, was inside the vesicles. And at that time, 14 years ago, we proposed the model where GXM was synthesized inside the cell. And using extracellular vesicles, the polysaccharide could go outside to form the capsule. So that was the beginning of this whole story. I'm not going into the details, but now we know that at least 20 different fungal species and approximately 60 isolates, the list of species are here, they are produced by fungal cells. So apparently this is something like a common physiological event used by fungal cells to export molecules in general. At the beginning, we are talking, actually, in the beginning, we are thinking about secretion. We were actually too concentrated in things that go outside, inside the vesicles. They could stimulate, for example, immune cells. They could be related to the delivery of virulence factors. They could be used for polysaccharide export, protein export, RNA export, and actually pre-release in fungal cells. But now we know that they come back inside and regulate a lot of different processes that are important for the physiology of fungal cells. So it is a two-way road. They go outside, but they come inside too. And the number of functions of fungal EVs is only growing and growing and growing. And several questions actually remain without any answer. And we are trying to explore this kind of questions. So right now, we are especially interested in two of these questions. Where do they come from and what they are doing in the outer space? This kind of question brings us to the second part of this talk, which is related to the science of biogenesis of fungal EVs. So for at least 7 to 10 years, we used the Saccharomyces model to explore the biogenesis of EVs in fungal cells. We tried at least 6 or 7 different genes regulated secretion. Fungal vesicles were always produced by mutant strains. So apparently, we could mess up, for example, with the amount of fungal EVs, sometimes with the composition, but we always had vesicles being detected in secretion mutants of Saccharomyces. We knew at the time that the composition was really complex. These proteins were actually part of a big network, and we knew actually too that they could be formed at the plasma membrane level, and they could be isolated from the plasma membrane and released into the cell wall. But we never had something really clear showing that fungal EVs could have their formation regulated by one classic secretion gene. So at that time, we started to suspect that the process was more complex than we thought at the beginning. So we started to look not only inside the cell, but at the plasma membrane too. And in this model, this was a model actually developed by Juliana Rizzo, which was a postdoc actually and a PhD student in the lab too. Now she's in the Pasteur Institute. And Juliana started a model investigating if fungal vesicles could be formed at the plasma membrane level too. She used this Aspergillus model, and she prepared what we called in fungal in the mycology field cells that correspond to protoplasts. They are cells artificially prepared to lack a cell wall. In this kind of model, we can take a look, for example, at vesicle formation at the plasma membrane level because you don't have the interference of the cell wall. So, for example, in protoplasts of Aspergillus, you could tell that you had something like vesicles being formed at the plasma membrane level. If you stimulate the synthesis of the cell wall, the number of particles resembling EVs, they increased in number, and you started to see fibers of the cell wall being formed here in association with the vesicles. If you take a look actually at the plasma membrane by fluorescence microscopy and scanning electron microscopy too, you can tell that the membrane can bud, bringing polysaccharides to the outer space. And actually, this can be confirmed here when you see the fibers of polysaccharides being formed in association with vesicle-like structure. So we started the usual protocols to isolate vesicles from Aspergillus. You can tell that they have the typical properties of fungal of eukaryotic vesicles in general. This is actually non-attracting particle analysis, and this is electron microscopy. This is fresh protoplast. I mean, you just remove the cell wall and isolate vesicles. And during the synthesis of the cell wall, you can tell that you have vesicles too. But the number of vesicles is different in these two different conditions. I'm going to show you this right now, but it's important because when you analyze the synthesis of the cell wall, you have a polysaccharide in association with the vesicles. When you don't have the stimulation of the cell wall, there's no polysaccharide. So this is an indirect evidence suggesting that fungal EVs could be related to the cell wall in fungal cells. Indeed, using different approaches, you can tell that during the cell wall synthesis, during the synthesis of the cell wall, you have more vesicles. So apparently there's a relation here. And if you take a look at the compositional analysis, you can tell that many of the enzymes important required for the synthesis of the fungal cell wall, they are associated with the Aspergillus vesicles. So this is actually a very short summary of a long story showing that fungal vesicles can be formed at the plasma membrane level too, and actually they are associated with the synthesis of the cell wall. And so in Aspergillus, EVs originates at the plasma membrane where they participate in cell wall synthesis. So I'm going to concentrate the larger part of this talk on the functions of fungal EVs. And I will start doing this with this model here that is Penicillium digitatum. This is actually a plant pathogen that can damage orange and other seeds too. You have a lot of economic loss because of the infection of orange, for example, and other fruits by Penicillium digitatum. And it's interesting because to cause this kind of damage here, penicillium produces a lot of toxins that go outside and they are deposited on the interface between the fungal cells and the plant cells. When they go outside, for example, they cause damage to the surface of the fruit, but they can actually inhibit the germination of seeds too. So apparently you have an important toxin that goes outside and causes a big mess in plant cells. So we hypothesized in collaboration with Thaisa Phil in the state of São Paulo in Brazil, that these toxins could be exported in vesicle soup. But at that time, we didn't know if this fungal species could produce vesicle soup. So we isolated vesicles from cultures, culture medium, but we isolated the vesicles from the in vivo model of plant infection too. They have the typical properties of eukaryotic vesicles. They have the size that is expected for other fungal species and a different range of eukaryotes too. They have the same morphology, and importantly, they contain the toxin too. So by mass spectrometry, we detected the major toxin, tripeptide A, produced by Penicillium digitatum, and they were actually an important component of fungal EVs. Of course, they are not the only one. You have a lot of toxins there, but when they are exported in the EVs and get contact with the seeds of oranges, they can cause damage to the seeds, and then you inhibit germination. So apparently, EV production, it is an important mechanism required for the export of toxins in a plant pathogen. And so EVs participate in the export of toxins that affect the plant host interaction in the penicillium model. So from now, I'm going to talk about Cryptococcus only. Once again, this is our major model, and I'm going to show you different aspects of EV production in this kind of model. So, of course, I told you about the beginning of the story 14 years ago, but recently, in collaboration with Mauricio Del Poeta in the US, I had a student, a PhD student there, Ana Carolina Colombo, who was checking if cryptococcal EVs could protect different hosts against a lethal challenge with Cryptococcus. So Ana Carolina demonstrated that in the invertebrate model correspond to Galeria melonella, which is an insect that reproduces the mammalian model of infection with different fungal pathogens. She demonstrated that EVs could they could protect, actually, at least partially, the host against a little challenge with Cryptococcus. So this is the regular curve of Cryptococcus killing the invertebrate host. And this is when the host received vesicles before the infection. So apparently there was a vaccine potential here. The manuscript was actually published a couple of years ago. But I want to highlight that one of the questions that we got from the reviewers was this one here. Can you demonstrate the same kind of thing in a mice model of infection? Of course, this is what we wanted to see. But at that time, the protocols to isolate vesicles were not really efficient at all. So, for example, to get a decent amount of vesicles to immunize mice, it was actually impossible because you need liters of cultures, and you could take forever to get a only decent amount of vesicles. It was a big problem at that time. So the protocols that we have been using at the time were the regular ones. We took supernatants. We tried to isolate vesicles from the culture supernatants, and then you can, for example, go for chemical analysis, stimulation of host cells, proteomics, and the regular stuff that everybody does in the field of EVs. But the problem in our model was that the yield was really, really low. So the amounts that we could get using the protocols was too small, for a big number of different experiments. And for mice immunization, it was not possible at all. So we started to think differently at that time, because if you think about nature and infection in general, you're going to realize that fungal cells, they are not usually in liquid matrices. For example, if you think about Cryptococcus, of course, they can go into the blood and then they invade the brain, the central nervous system. But in general, they are not in liquid matrices, they are in tissues. Aspergillus is the same. If you think about the environment, it is the same too. In general, you don't find fungal cells in liquid matrices. So because of this situation, Flavia in the lab here in Curitiba, she asked if you could isolate actually EVs from solid cultures. What is the advantage of this kind of protocol? You can combine a lot of different plates into a single tube, and then you go for the centrifugation with a very controlled volume of fungal suspension. So if you take a look at this video here, that's the idea. You can scrap the cells from the plates, and then you suspend the cells in a single tube, and you go for the regular centrifugation process. For example, instead of using, in our case, 2 liters of cultures, you can combine, for example, 5-6 plates into a single tube, and then you have 30 ml to isolate vesicles. So the time that you spend using this kind of protocol is much, much smaller. And then we started to have a lot of vesicles. And this situation, this kind of protocol changed the situation completely. So yes, you can have vesicles using the solid medium protocol. Apparently, the protocol is really reproducible. You can change the media, you're going to find vesicles there. You can use different fungal species, you find vesicles there. But the difference is that instead of taking 2 weeks, which was actually something regular in our model, you can have vesicle, for example, in 5 hours. We have been trying this protocol under different conditions, and apparently it is really reproducible and really fast too. So we started to use only the solid medium protocol in our analysis. And for example, because of this facilitated protocol, Bianca and Luisa in our lab, they observed, for example, that fungal vesicles can be really diverse. So what I'm trying to say here is that, for example, we spent many years using a single isolate, a couple of mutants, another species, and we have this impression about the properties of vesicles. If you have a better protocol, for example, instead of using two strains, you can use, for example, 15 at the same time. And then you have a better capacity to analyze, for example, if they're all the same or if they are different. And if you take a look at, for example, at these results here, you can tell that different isolates of Cryptococcus, they produce vesicles with different properties. For example, these three here, they are enriched with vesicles of larger dimensions. So this is actually really tricky because different properties lead to different functions. And if you think, for example, if the standard isolate of Cryptococcus neoformans, which is this one here, represents the whole genera, you can actually go into a bad situation because they're completely different if you increase the number of isolates. And because of this new protocol, for example, we started to learn basic things that we have not been able to learn before. Now we know, for example, that the peak of EV production, it is 24 hours after inoculation of fungal cells in the medium. And we know, for example, that larger vesicles, they decrease with time, while small vesicles increase with time. So apparently you have a regulated system here that implies that vesicles of different origins can be produced at different stages of the culture. I'm going into this very quickly. I'm not going to bother you with experimental details, but for example, with this kind of new protocol, we learned that different fractions of the a centrifugation gradient, for example, can be enriched with vesicles of larger sizes. And in our case, this is apparently important because the big vesicles apparently are the ones that contain the capsule polysaccharide. And this is actually important for capsule construction because we know that mutant strains enriched with this population here of larger vesicles, they are more efficient in producing extracellular polysaccharides, and they build let me show this one, which is clear, and they build a capsule more efficiently. So apparently there's a connection between larger vesicles, capsule construction, and the efficacy of capsule construction in fungal cells. So we have this question here, if this polysaccharide is really linked to larger EVs, and apparently it is, because if you stimulate the synthesis of the capsule, you have more vesicles of larger sizes. So apparently there's a connection between larger vesicles and polysaccharide export in the Cryptococcus model. And I'm not going into the details because this is old data, but apparently this is consistent with a study that we developed some 7 or 8 years ago, showing that if you delete a regulator of proteins related to the SNF7 pathway of RIN1 activation, you have a decreased capsule size and you have a decreased virulence too. So apparently you have connections here related to the biogenesis of fungal vesicles and the export of polysaccharides. I'm going to focus on the composition of the vesicles because, once again, because of the new protocol, we have been able to study molecules that we haven't been before. So with a lot of efforts, we had already described the composition of cryptococcal vesicles related to RNA, proteins, and lipids. But we knew nothing about small molecules. We had problems of sensitivity, reproducibility, so we had nothing conclusive. When we started to use the solid medium protocol, and the situation changed. So this is a general outline of the kind of analysis that we do to make sure that we have small molecule components of vesicles there in the Cryptococcus model at least. So it has to be present in 3 independent triplicates. It has to be present in vesicle fractures, but in cellular and supernatant extracts too, because it comes from the cell and it goes outside. It needs to be absent in control samples. And then you have to structurally validate this component using synthetic models. So, and then you go for mass spectrometry. So if you can replicate the kind of spectrum that you have with native vesicles following these 3 steps here, and the pattern is the same, then we consider that we have a vesicle component here. This is something that we have been doing with Taisia too, and we found different molecules there. I'm going to focus in one of them because we already knew, in collaboration with Guillaume Jambon in the Pasteur Institute in France, a collaboration with my former students and postdoc Juliana Rizzo too, we knew that that vesicles could be important for the survival of mice infected with Cryptococcus. This is actually true for Candida too, another important fungal pathogen. You could protect mice against a lethal challenge with Candida if the mice received vesicles too. But the components related to protection were not known. Actually, they're still not known. In our model, we found different molecules, small molecules in the vesicles, but this is just an illustration of the model that was actually describing to you a couple of slides ago. So actually, this is a synthetic standard, this is the EV extract, and this is the control. So the synthetic and the native extracts, they should be similar, and it should be absent in the control too. So this is the kind of work that we do to make sure that we have a vesicle component there. It's a lot of work, but then you make sure that this component, it is actually a vesicle component. We tested the functions of many of them, but one of them, this tripeptide here, isoleucine, proline, isoleucine, which is actually following the validation that you use for vesicle composition. This one was really special because if you give the peptide together with crypto to the invertebrate host Galleria melonella, you can actually protect the host against a lethal challenge with different species. I'm illustrating here with one of the pathogens, Cryptococcus gattii, but the result is the same with Cryptococcus neoformans, another major pathogen in this genus. So apparently, this is a very efficient peptide, which is able to actually rescue Galleria melonella against lethal challenge with Cryptococcus. So the survival curves are really clear, and actually you have less fungal cells in infected Galleria that received the peptide. So apparently, we don't know the mechanism because this peptide is an important inhibitor of a major enzyme related to fungal pathogenesis. But actually, Galleria defends itself against fungal infections because of innate immune mechanism. So we speculate that the peptide could be related either to immunological activation or inhibition of fungal enzymes that could be required for fungal pathogenesis. But this was actually the first component, the first EV component in fungal cells related to protection against a major pathogen. So the conclusion here is that more efficient protocols, at least in the Cryptococcus model, reveal that EVs are highly diverse and they can carry small molecules with an important immunological potential. So to finish, this is a little propaganda here, because if you are interested, for example, in fungal EV molecules, and if you're interested, for example, in making mutants, and if you're interested in studying the specific function of one protein or another, I need to let you know, for example, that we still don't have biomarkers for fungal EVs. You can visit, for example, the data bank that we developed with my collaborator, Fabio Passetti, here in Fiocruz, and you're going to find here a database where you can search for proteins that are present not only in Cryptococcus vesicles, but in any vesicle described so far. Of course, the vesicles in which the proteomic composition was investigated. So then you can, for example, think about vaccines, making mutants, studying biogenesis. It's really useful. We have been using a lot. And I recommend you, for example, to visit this data bank to take a look at vesicle components here. It was just published, it has just been published a couple of weeks ago. So this is fresh stuff. But I think the take home message here is that the biological potential of fungal EVs is really unpredictable. We still have a lot to know, to learn about this kind of structures here. Compared, for example, to other models, it is really, it's in infancy. And actually, we invite people to come to this field to generate more knowledge, because we believe that there's fungal EVs, they're important for a number of different processes, and maybe there's a potential here, for example, for drug development and vaccine development. And I believe there's a lot to learn about this kind of structures in fungal cells. So I need to acknowledge the collaboration of different people in different parts of this story that started 14 years ago, a lot of different groups in Brazil. This is our lab in Fiocruz, and these are our collaborators in Brazil. If you take a look, for example, at different publications, all of these people here helped us a lot in the understanding of how vesicles can be important in fungal cells. This also involves our collaboration with different groups in the US and in France too. So I need to acknowledge that a lot of people helped us to learn a little bit about fungal vesicles in general. And I really appreciate this opportunity to share the data that we have been generating in these years with you. I'll be open to any question now and maybe later. And once again, thank you so much for this opportunity. Carolina, thank you so much.
Okay. Thanks so much, Marcio. It's amazing talk and a wonderful presentation today. So I would like to invite everyone to write down their questions in the chat box. And if you also have some follow-up questions, you can also raise your hand and I can get back to you. So I think there's a couple of things that's really intriguing for me. So with the cell wall model or cell wall-less model, so is there sort of a conclusion on how the EV came across this barrier? Is there some sort of like cell lysis needed? What do you think about it?
That is actually a major question in the field, because we still don't know. So I was showing an early step suggesting that, for example, in Aspergillus, vesicles could help with the synthesis of the cell wall. And you're asking actually a big and important question, that is, how do the vesicles cross the cell wall? So, for example, in the cell wall of fungal cells, you have nothing like, for example, a pore, for example, actively bringing the vesicles outside. You have nothing like this. But in the vesicles, you have a lot of enzymes that have the ability to hydrolyze cell wall components. So one hypothesis is that fungal EVs can actually auto-catalyze their passage through the cell wall by hydrolyzing cell wall components. For example, in the composition of the cell wall in fungi, you have a lot of glucan there, a polysaccharide made of glucose. And there's a lot of glucanases, for example, in fungal EVs. So maybe they're actually hydrolyzing the cell wall and promoting their passage. It has not been demonstrated. Another possibility is that you have an elevator mechanism. Let's say the cell wall was just, cell wall synthesis just started, and you have a very thin layer of the cell wall and vesicles being produced at the plasma membrane level. When the cell wall is being synthesized, the cell the direction of synthesis of the cell wall is from inside to outside, and then you can bring vesicles over a cell wall layer until they go outside. So this is actually has not been demonstrated either, so it's only a supposition. But now we know, for example, that you have a transit of lipid structures across the cell wall. Neil Gall, it's a very important name in the field of medical mycology. He demonstrated a couple of years ago that artificial liposomes containing antifungals, they cross the cell wall in their intact shape. So whatever the hypothesis is, whatever is the mechanism, whatever the mechanism is, The point is that vesicle structures, they can cross the cell wall in their intact form. And actually 20 years ago, we saw lipid structures there, membrane structures there that appeared to be in their native form. So the point is, they go there and they go outside, but we still don't know how. So this is all hypothetical and it has to be demonstrated.
Yeah, I wonder with that capsule that is protruding, is there actually cell walls surrounding the capsule? Or would that be like one of the exit point for the EVs to go outside?
Yeah, it's possible too. It is possible too. So when fungal cells, when they divide, they have to hydrolyze the cell wall to separate. So to synthesize the cell wall is of course important, but to hydrolyze the cell wall, it's important at the same level. So for example, this is actually a nice hypothesis. For example, when you separate cell walls and then you hydrolyze the cell wall, then you have a specific site from which vesicles could leave and go outside. So I think it's possible too.
Yeah, that's very interesting. So we've got a few questions here, so I'll go back with my question later. But Donovan, could you unmute yourself and start the video?
Yes, thanks, Carolina. Can you hear me there? Thank you, Marcio, for a great talk. As you mentioned, it's good to have these spaces for researchers on fungal EVs. We're based in Melbourne, Australia, and we follow your work closely. So thanks for a great talk. My question is regarding the production of the Penicillium toxins. If you have any evidence or any hypothesis as to where these toxins are produced inside the cell and how they might be targeted to EVs for subsequent export. Thanks.
Thank you, Donovan. Thank you for your words. And yeah, so this kind of toxins, they are actually the secondary, they're actually the rest of metabolism in general. So there are secondary products that are formed after, fungal cells are doing their regular physiology. And actually they're really toxic in general for fungal cells. So they need to go outside. And when they go outside, they can actually be toxic for human cells and plant cells. So that's the classic functions that they have. But to target molecules into fungal vesicles, it's a big gap in the field. So for example, just to be straight, I don't know, but if you think of, for example, about RNAs, it's really intriguing because we don't know how they go there in fungal cells. But if you compare, for example, the distribution of RNAs in the cells and outside the cells, you can tell that the distribution is completely different. You have, for example, some molecules that if you have a hypothetic scale, they are 1 inside the cell and they are 100 in the vesicles, and the opposite too. So apparently, I spent many years thinking about garbage, which is something normal in any kind of cell. For example, when a cell have to regulate the cell volume, you need, for example, to get some pieces of the membrane out. And when they go outside, they contain, for example, biologically active molecules, and then they can, for example, to stimulate a host cell. So the function is actually noble, but the mechanism of biogenesis is something like, for example, throwing garbage out. But that seems to be not the case, because if you think about the RNA model, you can tell that there's something directing RNA to the vesicles that do not reproduce what you see inside the cell. And for the toxins, for example, we still don't know. Actually, the presence of toxins in vesicles was not something that we were sure about a couple of years ago. And we actually don't know how they go there, but that should be something regulated. But just to finish and to wrap up, one advantage of packing these molecules inside vesicles and throwing this out is, for example, that you protect the cell against the toxicity that they might have. And if you think about, for example, about the interaction with host cells, you concentrate this kind of components in a very small bag instead of having these things floating around in relatively low concentration. So that should be advantages, but the mechanism of packaging, we don't know at all.
Maimona, you have follow-up question?
Yeah, I have a question. Thank you so much for the impressive talk. Actually, I'm working with mammal cell-derived AVs, so it was very interesting to know about fungal AVs, something new for me. And I'm very impressed actually with your idea of solid medium protocol for isolation of, for culturing the cells and also isolating EVs. I hope something similar will be developed for us with cells, because we are working with massive amounts of, of medium and all. So yeah, my question here, so when you isolate EVs, are they considered as released EVs, or they're still contained in fungal cells in that case?
So thank you. I like this kind of question because when we started the new lab, I don't do nothing. So I just write things. So people that do the work, they have to learn the techniques again. So we had someone, Juliana, from the old lab coming to the new lab to teach people here, to isolate vesicles. And Flavia, who was learning the protocol, the classic protocol, she said that, this protocol is a nightmare. We need something better than this. And the idea of getting, using the solid media came from this kind of situation. And she asked, so do you think it's possible to have vesicles being produced in solid media? And if you think about physiology, it doesn't matter. If it is a process that is important for cellular physiology, and if the cell grows in solid media or liquid media, it doesn't matter. So if it's important, it's going to be produced in any type of media. So that's how we started to use the solid medium. And the solid medium and the liquid medium, we always got the same kind of question that you're asking now. At the beginning, we didn't know, so we need actually dozens of different methods to prove that this kind of structure was released only by living cells. So to make a very long story short, we tried, I don't know, a lot of different approaches to make sure that we could detect them only if the cells were alive. And at least for Cryptococcus, so people that started work with other species found the same thing. But with Cryptococcus, for example, after a couple of years, we were sure that there were products being released by living cells. So I think now we can assume that that's the kind of condition that we are working on. But it took a lot of time. I have to tell you that the first paper describing EVs in Cryptococcus was rejected 6 times, only because of these technical issues. And I think it was normal and acceptable. It was something new. People were not sure about it, so they had to ask. Actually, it was important for us to be sure that we were not working with artifacts. But at the beginning, this was the kind of question that we had to face all the time. But it was good because now we are pretty sure that they are released from living cells and not from other conditions.
Thank you.
Apologies to Siba Sensei. Please go ahead asking your questions.
Thank you very much. Great talk. I was impressed with your tripeptide experiments. So how did you select the peptide? Is that a part of a protein or part of a toxin, something like that?
Yeah, it is a good question because we don't know. So we found there in the vesicles the free form of the peptide. But of course, it could be cleaved, for example, from a larger protein. It could be associated to other kind of molecules. It could be a secondary product of the metabolism in general. So we don't know. We know that it was there in the free form. We found vitamins there too, pantothenic acid, and other small molecules. But this one is interesting because it is an inhibitor of an enzyme called dipeptidylpeptidase 4. And this enzyme is involved not only with the pathogenesis of fungal cells, but in insulin metabolism and recruiting of immune cells. So yeah, maybe it could be just free in the cell being exported for any reason, but I don't discard the possibility that this peptide was cleaved from another protein or any other kind of molecule, and then it has a primary function in cell physiology that is not related to immunity or pathogenesis. We have to keep in mind that the primary niche of this organism is the environment. For example, Cryptococcus is in the soil. So the peptide was not there to protect, against any immune cells. It was just part of the regular physiology. And yes, it could be cleaved from a larger molecule. And I don't know, it's a good question, but we don't have the complete answer.
Thank you very much.
Augusto, please go ahead. Thanks for waiting.
Thank you very much. Great talk, Marcio. Very speculative questions. The first, would you think about the role of this EV on cross-kingdom communication? And the other one is, would the vesicles have signatures? I mean, the EV from crypto would be different from the EV from Aspergillus, for example.
Thank you, August. August was supposed to be my friend, so I was expecting simple questions. But yeah, so in terms of cross-kingdom communication, it has been demonstrated, in a beautiful paper from a couple of years ago. So you can have, for example, plant EVs going inside fungal cells where they regulate gene expression. So in this case, actually, they'll neutralize the expression of virulence-related genes. So it was a beautiful and sophisticated mechanism of neutralization of virulence, and has been published in Science 2 years ago. But the opposite is not clear yet. So fungal cells producing, for example, vesicles, going inside other organisms and, for example, influencing the gene expression or the physiology in general. So if you think about immunology, yes, because, fungal cells, they produce EVs that stimulate the immune response in mammalian hosts. This is actually a kind of interkingdom communication. But this is actually a consequence of fungal physiology during infection. But something regulated like they demonstrated for plant and fungal cells, it was not demonstrated when you have fungal cells producing vesicles and going outside. So I think this is actually very hot in the EV area, especially for fungal cells, and it has to be demonstrated because the impact of this kind of phenomenon could be really, really huge. And as for the markers of vesicles in different organisms, it is a good question too, because if you think about classes of molecules, you have a lot in common in different fungal species. For example, you have a lot of proteins that are related to cell wall hydrolysis, sugar metabolism, RNA binding proteins. In all kinds of vesicles in fungal cells, you can find this kind of structure there. But if you go to the database that we generated with Fabio Passetti, you have only one molecule that is present in all fungal cells studied so far. And you have a few heat shock proteins that are present in most of the fungal cells too. So it is a big challenge in the field to find biomarkers of fungal EVs in general and biomarkers that are specific for some species of fungal cells. I was expecting more than one protein for all vesicles there, but actually there's only one so far, and we need to look for more. So there's a preprint, a paper from Guillaume Jambon that is available as a preprint showing that monoproteins are important in cryptococcal vesicles, important components. They are present, for example, in 97% of the vesicles in Cryptococcus. But it doesn't mean that they are not present in other vesicles too, because it has not been investigated. But apparently they are important components. And this is actually too important because, for example, you can design novel techniques to isolate vesicles, for example, using lectins that can recognize monoproteins. So you can actually develop new protocols. So this is an open field inside the field of fungal EVs, and I think there's a lot to learn. Biomarkers are actually unquestionably important, and we need to spend time to understand this a little better than we do now.
Thank you very much.
James, please go ahead with your question.
Yeah, hi, great talk. I work with Donovan. So I'm just interested in interrogating your hard media isolation method. And I was wondering how the yield of EVs compares per biomass, or whether you've even looked at it per biomass on hard media versus liquid media.
Yeah, good question too. We haven't compared. So after we started to work with the solid medium protocol, people in the lab, they run away from the liquid protocol, because in Cryptococcus it was really, really difficult. So, and at that time when we were using the liquid protocol, we still didn't have the nano-tracking particle analysis machine. So, but we have an idea in terms of the number of vesicles produced per cell. Cryptococcus is unicellular in liquid and solid media, and apparently it is not that different. We are talking about, so one thing that we need to keep in mind is that you lose a lot of vesicles during isolation, not only because you just lose, but fungal vesicles, and in the case of Cryptococcus, it is using the vesicles to produce the capsule all the time. So we're not talking about a real number. We are talking about the number that we are able to detect. And we are talking about 2 up to 5 vesicles per cell. And if you go straight to the liquid medium, the numbers are not really different. So apparently it is more a matter of time that you save than more vesicles that you get. But of course, if you can combine many cells into a single tube, it is much more efficient, for example, than centrifuging 2 liters of cultures. That was the case until 3 years ago. So, but I think that in terms of physiology, the number of vesicles per cell is not really different. It's more a matter of time than the number of vesicles that the cells produce.
So I think following up to that, how different is the cargo profile compared to the liquid?
So it has to be determined, because the only fungal species that at which the composition of vesicles was performed in solid and liquid medium was Cryptococcus, but the conditions of analysis were totally different. The machines that were used for proteomics analysis were totally different too. So this is something that really needs to be updated. And so apparently it's completely different, but the conditions of analysis were completely different too. So I would say that it has to be repeated under the same conditions to make sure we have something comparable. With the data that we have now, I don't think it's possible to compare.